Okay, so what I'll do is also there's no audio in another slide, and what I'll do is also expand the yeast to hybrid system because there's something that I missed talking about, and that is um, how we make a library. Okay, so let's start with protein-protein interaction. Now remember that proteins again cannot or do not act by themselves. They have to interact with other molecules, whether it is a metabolite, small molecule, large molecule, carbohydrates, lipids, other proteins, and so on. So protein-protein interaction is very important for cell function, and it's really of interest to know what proteins interact with other proteins. Okay, that we can understand mechanisms and we can design drugs against this interaction. There are two methods that I'll talk about. The first one is known as immunoprecipitation or co-immunoprecipitation. The idea here is that whenever you hear the word immuno, you know that we're talking about an immunoglobulin or an antibody, which is a protein that is produced by our immune cells to bind to and fight antigens, foreign bodies. Now, one important characteristic of immunoglobulins or antibodies is that they do interact very, very, very specifically to proteins. Okay, so we can design or we can produce antibodies against certain proteins. And there are different ways, and I'm not going to talk about this. So the idea here is that let's say that I take a sample, cells, bunch of cells, and I open up these cells, and these cells would release all of the proteins inside them. Okay, so I have large number of proteins in my samples taken out of cells. Now, notice that since proteins interact with other proteins, you would have what is known as protein complexes. So here you have a complex of proteins bound to each other. What we do is that we add an immunoglobulin that is specific to a certain protein. In this case, you can see that it binds very specifically to this purple protein. And we precipitate, we precipitate all of the protein complexes that are bound to this immunoglobulin. So we are specifically isolating or purifying the proteins that interact with our protein of interest, the purple protein. Okay, and we remove all of the other proteins that are not bound. We remove all of the other proteins that are not bound to our complex or our protein. So we take these proteins and we can release them from the immunoglobulin, and then we can analyze the nature and identities of all of these proteins using different techniques. An example is Western blot. Okay, so the idea here is that what I'm trying to do with this experiment is I'm isolating proteins that interact with a certain specific protein in order to identify what these proteins are. Okay, so this is co-immunoprecipitation. Co يعني مصاحب. Okay, so now the other technique, before I go into the yeast to hybrid system, there's something that I need to introduce, and that is the concept of a DNA library. So the idea is, what is a library? A library is, as you know, it's a building. It contains shelves. You go there, different floors. You're looking for a certain book, so you search in the computer, and it tells you it's this book is present on in the second floor, aisle number, I don't know, 30, shelf number 45, and you can then go to the second floor, aisle 30, whatever, shelf 45, and you can take the book that you want. Same thing with the DNA library. So basically, it is bunch of plasmids. Each one of them contains a certain DNA insert. And we keep these plasmids either inside bacteria or as purified plasmids, and we keep them in the freezer. So whenever I want to study a certain insert or gene, let's say, All I need to do is that since everything is organized, I go into the computer, I say I want to study this particular insert or this particular gene, and it tells me, well, go to freezer number 2, shelf number 4, box number whatever, 95, and your vial that contains the plasmid or bacteria would be present in this particular position. So I take it and I study it. Okay, so it's the same idea as a library. So what we do here with a DNA library is that we have different clones, okay, of or different plasmid molecules, each one of them containing a specific DNA insert, that and I know what this insert is usually. Okay, so how do we create a library, a DNA library? There are two types of libraries. The first one is known as a genomic library, and the other one is known as a cDNA library. A genomic library is basically, we take a whole genomic DNA, the whole DNA of a human cell or cells, okay, and we fragment them into smaller pieces, and we place each piece into a plasmid. So now each plasmid contains a specific piece of DNA, okay? And then we take these plasmids and we put them inside bacteria. Now each bacterial cell would contain a plasmid, and each plasmid contains a specific insert. And we grow these bacteria individually, or on dishes. Each bacterial cell would form a colony, and we can then take these colonies one by one, and we can grow them further, freeze them, and we can identify what bacterial cell contain what plasmid and what insert. Okay, so that's a genomic library. Now the other library is known as a cDNA library. C stands for complementary, and a cDNA is made of mRNA, not DNA. So the idea is that we take messenger RNA, and this messenger RNA, in human messenger RNA, is fully processed. So it does not have introns. It's only exons connected to each other. We convert the messenger RNA to cDNA using reverse transcriptase. Okay, we talked about that before. Now, so what we have here is a piece of DNA that represents an expressed gene, all right, with no introns, just exons, full-length genes. And we take each cDNA and we put them in a plasmid. Now, each plasmid now contains a gene, okay? Now, we, again, insert these plasmids inside bacterial cells. So each bacterial cell would have a plasmid, and each plasmid would have a gene, and we grow them the same exact way. We grow them on dishes. We take colonies representing specific clone of cells, okay, and we can freeze them or do whatever, and we create our own library, DNA library, in a freezer. Okay, so whenever I want to study a particular gene, I would go into the catalog, and I would again search for where this gene is located. That is, in which vial containing which bacteria, which shelf, freezer, lab, whatever. Okay, so that's the whole idea. This is another representation of the different types of genomic and cDNA libraries. Now remember, with the genomic library, it's much more complex than a cDNA library, because here with the genomic DNA library, we have everything. We have introns and exons and promoters and enhancers, coding regions, non-coding regions, regions representing just RNA genes for RNA molecules, or for genes that would code for proteins. We have telomeres, centromeres, bunch of stuff, everything in a genome. So it's very complex. So it's much better to do a cDNA library. Again, messenger RNA gets converted to cDNA, representing a full gene made of exons, no introns. We put each gene into a plasmid, and we insert each plasmid inside a bacterial cell, and this bacterial cell would grow into a colony, and so on. So now we have a library of genes right now. Okay. Okay, so let's talk about the yeast to hybrid system. Now, this technique relies on taking advantage of domains. Again, what is the definition of domain? Domain is part of a protein. It's a three-dimensional structure, okay, that folds independently of the rest of the protein, so that it takes up its own structure independently of the rest of the protein. So if we isolate it, if we cut it off a protein, it can still fold, it can still take its own structure, and it can still be functional as well. Okay, so this is the important part. So the yeast to hybrid system relies, or it's based on a genetic system that is based on, or that is activated by an element known as an upstream activating sequence. Okay, so this upstream activating sequence it binds to a protein known as GAL4. Okay, right here. So this is GAL4. Now GAL4 is made of two domains: a DNA binding domain and an activating domain, or an activation domain. Okay. Now once you have binding of the protein into the onto the UAS, you have the activation domain interacting with the RNA polymerase that sits on the promoter, activating the RNA polymerase and starting transcription of the gene. The gene that is in here is actually a bacterial gene. So this is the genetic system that is in use. So it's a bacterial system. It's a bacterial gene. It is the LAC-Z gene. It's the one taken from the lac operon that produces the beta-galactosidase. And it's known as a reporter gene, and I'll tell you why it's known as a reporter gene. Okay, so what happens here, so this is really a fancy system because you have a yeast genetic system with a bacterial gene, and you can also insert human genes as well into these yeast cells. Okay, so the idea now is that we have a protein known as GAL4. It's made of a DNA binding domain, activation domain. If you have binding to the UAS system, you have induction of transcription of the LAC-Z gene. So why LAC-Z? Well, it's a nice enzyme, beta-galactosidase, and what it does is that it cleaves lactose into its components. Okay, except that here what we do is that we're not using lactose, rather we're using a specific metabolite known as, or substrate known as X-gal. It looks like lactose, except that it has this group right here. Okay, so what happens is that beta-galactosidase cleaves this moiety, this group right here, so it becomes free, and it can be converted into something else that gives a blue color. So if a yeast cell produces beta-galactosidase and we add X-gal to these yeast cells, what would happen is that these yeast cells would look blue. On the other hand, if yeast cells do not express beta-galactosidase, what happens is that yeast cells would grow onto a dish like this one here, but colonies would look white, not blue. So you can see a combination of yeast cells, blue and white. So the blue ones express beta-galactosidase, okay, and they convert the X-gal into this blue precipitate or blue product. Okay, now and why is it that beta-galactosidase enzyme produced? Because the gene is active. The LAC-Z gene is active. Why is it that the LAC-Z gene active? Because the GAL4 binds to the UAS element. Okay, now what we do now is the following. This is what's really sophisticated in here, is that we make a plasmid, and this plasmid contains the gene X, which is a known gene. We know what gene X is, but when it is produced, when it is synthesized, it's produced along with the binding domain only of the GAL4 protein. So we have protein X, and linked to it we have the DNA binding domain. If we have this plasmid inserted into yeast cells, what would happen is that the DNA binding domain would bind to the UAS element, right? And X would be there as well. Good. But there would be no induction of transcription. Why? Because there is no activation domain. Good. And then we have other plasmids. We have a library of this other plasmid. Okay? Different plasmids having different Y genes. So we have, let's say, Y1, Y2, Y3, and all of them. So each gene is in a plasmid, and each gene, when it is synthesized, it is synthesized along with the activation domain of the GAL4 protein. So we have protein Y along with, or conjugated to it, or part of it, is the domain of GAL4, the activation domain of GAL4. So if we have this plasmid only inside these cells, you would have Y with the activation domain, but there again would be no induction of transcription. Why? Because there is no DNA binding domain. Okay, remember that we have different plasmids with different Y genes. So each yeast cell would produce a different Y gene. Now, what we do is that we insert both of these plasmids, so each into yeast cells. So we have yeast, every year's yeast cell would produce X, the protein we know, and would produce a different Y gene. Y1, another yeast cell would produce Y2, a third would produce Y3, and so on. Okay? So we have lots of yeast cells expressing X, and each one of them is expressing a different Y. So, the idea here is that if X, which I know, interacts with Y, which I don't know, then the binding domain and the activation domain are now close to each other, and you can have the activation domain interacting with the RNA polymerase, inducing transcription of the reporter gene. Okay, so we call it a reporter gene because if we add X-gal, the yeast cell becomes, or the yeast colony becomes blue. So what this gene is doing is that it's reporting to us something. It's telling us that there is interaction between X and Y. If there is no interaction between X and Y, it means that these two domains are not close to each other and there is no transcription, and as a result, yeast cells would look white. So here we have it. Then we have all of these yeast cells. Some look blue, others look white. What we do, so we are interested only in the blue yeast cells. So we take this colony right here, okay, and we grow it up, and then we release the plasmids, and we see what the identity of Y is. We determine what the gene is that produces whatever protein that interacts with X. Okay, we take this yeast cells, this colony, we grow it up again, and we release the plasmid, and we see what that gene or we see what the gene is that produces the protein that interacts with X. It would be a whole different protein, by the way, than the protein that interacts with X in this colony, and different than the protein that produced from this colony, and different than the protein produced from this colony. So what we do is that we collect all of these blue colonies, okay? We release all of the plasmids from them, and we identify what the genes are. We don't care about the white colonies because they produce proteins that do not interact with X. Okay? How do we identify the genes? Well, that's next lecture when we talk about DNA sequencing. and this is the yeast to hybrid system.